our mission is to support medical countermeasure development. So that would include both vaccines and therapeutics. We invest in basic research through applied and advanced research. So this includes discovery through preclinical development, submission of a package, an investigational new drug application to the US FDA, and then also we do some first-in-human clinical evaluation. In terms of the types of pathogens that we focus on, historically the Department of Defense, the Chemical and Biological Defense Program, has focused investments on countering defined pathogens. So essentially a list of known biological threats. And this list was generated based on several factors, but the most, I would say, primary factor is the potential to pose severe health and operational risk. However, as we saw with the COVID-19 pandemic, the expansion of both technological advances, but also the threat landscape has greatly increased. So this really, includes the emergence of novel infectious disease pathogens. And the challenge that we find is predicting when and where those new and novel pathogens might arise is the primary challenge there. And so a couple of years ago, the Chemical and Biological Defense Program took a hard look at how we're developing medical products. And in order to better prepare the joint force against both known and unknown threats, these would include naturally occurring pathogens and possibly engineered pathogens, we're moving away from viewing the threat landscape as a defined list and rather moving towards a focus on removing or reducing the impact of exposure to a particular pathogen. And so this shift demonstrates how we respond to product development in the future. And so that response is a two-pronged approach where we will be investing in broad spectrum or non-specific medical countermeasures, but an increased emphasis in establishing capabilities that will allow us to rapidly respond to particular threats as they arise. So our mission is to support medical countermeasure development, so that would include both vaccines and therapeutics, by investing in basic research through applied and advanced research. This aligns with the FDA approval process to include discovery through preclinical development, submission of an investigational new drug application to the US FDA, and also first-in-human clinical evaluation. So in terms of the types of products or pathogens that we focus on, historically the Department of Defense has focused on developing medical countermeasures that counter a defined list of known biological threats. And this list was generated based on several factors, but primarily focusing on the potential to pose a severe threat and a severe health and operational threat. However, rapid evolution of technology continues to drive the expansion of the threat list. And as we saw in the COVID-19 pandemic, the threat landscape now includes emergence of novel infectious disease pathogens. However, predicting when and where those novel threats might arise is extremely challenging. So to better prepare the joint force against future unknown threats, and this would include naturally occurring emerging pathogens, we're moving away from viewing the threat landscape as a defined list of known pathogens, and rather moving towards a focus on removing or reducing the impact of exposure to a pathogen. And so this shift demonstrates how we'll view medical countermeasure response as a spectrum that requires investing in the development of broad spectrum or non-specific medical countermeasures and establishing capabilities to rapidly develop narrow spectrum solutions for narrow spectrum medical countermeasures. So I mentioned this two-pronged strategy. The approach entails delivering non-specific medical countermeasures and also establishing capabilities that allow for rapid development of specific products. Investments in 2024, they're going to continue to focus on development of products that have broad application. So for example, broad-spectrum antivirals, immune modulators that have efficacy against a range of pathogens. And one example that I want to highlight is our investment in molnupiravir. So molnupiravir is a broad-spectrum antiviral. It was originally developed for encephalitic alpha viruses, but was later used for influenza and SARS. I'm also very excited about increased investments and capabilities that will enable us to respond more quickly to emerging pathogens. And this is where the Domain and Raptor programs come in. And so these are two programs that I want to highlight here. So the Discovery of Medical Countermeasures Against Novel Entities, or Domain, is an independent but coordinated parallel development program. It includes multiple platforms that are focused on identification of medical countermeasure mechanism of action, target discovery, and identification and evaluation of the actual medical countermeasures in both a preclinical and clinical setting. And these are the cornerstone components of Domain. And as they come online, the second stage is to integrate them into an end-to-end product development platform. And so, for example, one of the components of Domain is the pathogenicity and toxicity forecasting using multi-organoid systems, or PATMOS. And PATMOS utilizes artificial intelligence and human organoid models, also known as human organ-on-a-chip models, to identify pathogen and toxin interactions, dependency, and response. And the goal here is to try to identify potential toxicities of a potential countermeasure before we get into the more expensive and labor-intensive preclinical studies. One of the challenges that we experienced during the COVID pandemic was that there were a lot of potential candidate vaccines in development. And it was really difficult for us to figure out what was the best one that we needed to prioritize development, which one had the highest likelihood of success. And so this is where the Raptor program comes in. So the Raptor program is the Rapid Assessment of Platform Technologies to Expedite Response, or Raptor, and it aims to develop a predictive capability for matching a specific bacteria, virus, or toxin with the most appropriate vaccine platform. And so recently we've established a consortium of researchers across multiple fields of study, including computational biology, virology, immunology, bacteriology. And these groups aim to develop machine learning based capabilities that will allow us to rapidly select candidate vaccine for development. Both the Domain and Raptor programs are examples of programs that are part of a larger push within the Chemical and Biological Defense Program called the Medical Enterprise Architecture. So the Medical Enterprise Architecture seeks to integrate capabilities that are already existing or new programs as they come online into a larger rapid response framework. The goal here is really to reduce time and cost associated with product discovery and development. Within the Chemical and Biological Defense Program, there are two separate organizations, and together we're responsible for end-to-end product development of vaccines and therapeutics. So we are the Joint Science and Technology Office, and along with our counterparts at the Joint Program Executive Office, we fund discovery, so basic research, all the way through clinical development and fielding of products. So, as I mentioned earlier, we have the mission to fund basic research, applied research, which correlates to discovery, preclinical, and early clinical development. So we're very interested in learning about products that are in development that have broad spectrum activity. And again, this aligns very nicely with our two-pronged approach, which focuses on broad spectrum solutions in addition to capabilities. And we're also interested in leveraging products that are being developed for other indications. So we have a very robust repurposing program. So in the past, for example, we've funded studies for label extension for approved antibiotics. We've also evaluated cancer drugs as possible antivirals. So as part of that second part of our approach, we're also interested in learning about capabilities that will allow us to increase the speed and efficacy or efficiency of drug and vaccine development and evaluation. So for example, we're interested in developing organoid systems. These systems allow us to more quickly discern potential drug toxicity. So this again ties back into our two-pronged approach. And really the goal here is to reduce potential impact of a pathogen exposure on the joint force. We need to be able to develop medical products quickly, and we need to get them fielded rapidly within time, cost, and schedule.